Okay, guys. Hey, welcome to this the second go at this podcast. I remembered to press record this time, so we're off to a better start than last time. Um, Second part of this standard here. I've reorganized this, so excuse this for saying 4.1. That should say 2.1 with one of our standards that we're looking at. That DNA and sometimes RNA is the primary source of information in biology. We're focusing here on DNA replication. It's an abstract process, so you need to spend quite a bit of time with this. Trying to get it on your own and understand it on your own, and of course we'll talk about it in class as well. I first want to bring up this idea called the central dogma and what the central dogma means, because we're going to be looking at it quite a bit here. Central dogma is the idea of going from DNA and then making a copy of DNA. DNA has to stay in the nucleus and going to RNA, this useful copy that we can make, and then having RNA translated into a protein. It's a complex idea. But we use this name here, the central dogma, as what's happening when we talk about going from DNA to an actually physically expressed trait. And then, of course, physically expressed traits can be acted upon by natural selection, right? So DNA replication is the first thing that I wanted to talk about with you guys. And in DNA replication, what we're looking at is DNA going to DNA. So like in the Prezi, and also on this previous slide here, we're saying, okay, we got this blue portion here where we're saying DNA is the input, but DNA is also the output. So it better be the same thing. We're just focusing on how DNA makes copies of itself right now. That's what DNA replication is. The other processes will come a little bit later. So DNA replication, it allows us to make very accurate copies, and for that information to be passed along from generation to generation. It happens semi-conservatively, which is something that you guys are working on right now. With the Pulse Chase primer, that resource from HHMI. You guys have time to work on that again tomorrow. Each strand has a template strand that then gets passed along to its new strand, in which there's a complementary copy on it as well. So you have this semi-conservative replication where the DNA needs to open up, it needs to be copied, and then there needs to be a final split where now this DNA strand has part of the original parent strand and this one also has part of that original parent strand. Okay, so that's what we mean by semi-conservative, and hopefully we nail that down in a little bit of time we have in class tomorrow. There's very complex things in this process called enzymes. Enzymes, for now, what I'd like you guys to think of them as are molecules, little packets or proteins that do a very good job with one thing only. So it's sort of like a worker on a line worker. If they're putting together a car, they might have one job that they do very efficiently and one tool that they use very efficiently in order to put a certain screw into the engine of the car. All of the factory workers working together, all of the line workers together are able to make something amazing like a car. In this case, we're trying to do something amazing, which is DNA replication, and that requires lots of line workers. Those line workers that do one job really well are enzymes. And there are a bunch of very complex ones here. So I want to introduce you to some of them, which ones you have to know and which ones you don't need to know. Helicase is one of the first ones here. So if we're looking at this DNA strand, what we're trying to do is make a copy of it. Helicase is an enzyme. If it ends in ACE, ACE, if it ends in ACE, it is an enzyme. That's how they're named. Helicase opens up and unzips this double helix that is DNA. It breaks the hydrogen bonds in between the letters, the bases, the nitrogenous bases of DNA. It opens them up and it exposes the bases now that no longer have their complementary partners. So here you can see this top strand in yellow. This bar here is representing bases like A or C or T or G. And then you have the complement over on this side in the green, which for A would be T. The helicase splits those open and allows the helix to be opened up. Topoisomerase, which is represented right here, this green sphere, it rotates the helix. So the helix we know is twisted and sort of crunched up into the double helix. What topoisomerase does is it rotates it around and allows it to go linear and straight. So that way, helicase is able to do its job, come along and unzip the bases in order to expose the bases. That's what those two are really good at. We'll get to a couple of these other ones. DNA polymerase, and if you read your book and you go through the Prezi, and I even might mention it a few times, there's different types of DNA polymerases. Just knowing DNA polymerase is going to work okay for us. DNA polymerase is probably the main player here because they have the main job in DNA replication of making sure that we have exact copies being made of DNA strands. So DNA polymerase, which is represented up here in this blob, is going to go along and make sure that we're matching complementary nucleotides with the nucleotides that are laid down in this parent strand. So right here, you can see we're working in a 5 to 3 prime direction, and that's how all enzymes are going to add new strands in a 5 to 3 prime direction. So this complementary strand, this parent strand here, you can see goes 3 to 5 in this direction. DNA polymerase is going to attach itself, and it's going to add in a 5 to 3 prime direction and start adding nucleotides. So for A here, this DNA polymerase reads and says, well, there's an A, so there must be a T nucleotide that's added to this, so it adds T. There's a C on this parent strand, so it adds G, so on. One of the interesting things as you look through the Prezi will be that these are how the bases come in the cell as nucleoside triphosphates. The one that you are most familiar with is ATP, adenosine triphosphate. So if there was an A attached to this, it would join it up, and then these two phosphate groups on the end of that snap off. This phosphate group then becomes part of the new backbone, which you can see is right here. Energy is released, and this energy is used to carry along the process of DNA replication. One very, very important thing to mention, other than going in a 5 to 3 prime direction, always, 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 is that DNA polymerase makes mistakes. It works incredibly quickly to add new bases to DNA. You have your genetic code is made out of 3 billion letters, and in every single cell, when your cell gets ready to split or ready to make new copies, it needs to copy all 3 billion letters of your genetic code. So mistakes are inherently made. Now there's backup for that. There's backup to ensure that that fixes either get made or that it doesn't have that big of an impact. But when you do have a change that's made during DNA replication, it's called a mutation. When you have a change in this letter, which we will get into the impacts of that later on in this unit. Again, something to mention, polymerase can only add, it's directional, it can only add in a 5 prime to 3 prime direction. Something that you should be able to do with DNA replication is say where the leading strand is and where the lagging strand is. The leading strand is nice. It is an easy strand. People understand what's going on. There's less enzymes involved. It goes in a straight direction, easy for the cell to do, to go through replication. The lagging strand is such a pain in the butt. It makes things incredibly difficult because this enzyme can only work in a 5 prime to 3 prime direction. So let's look at that a little more closely. We've got this model right down here. One is labeled as the leading strand, the other is labeled as the lagging strand. One thing I'd like you guys to be familiar with, and you'll see this in the Prezi, is things like eukaryotic chromosomes, R chromosomes, are going to have multiple bubbles, replication bubbles, that form in the chromosome. And then when you form a bubble that opens up like this, there's replication forks, where this would be a direction of the replication fork, and this is. This right here is also a good example where this helicase, this blue triangle is sitting, would be an example of a replication fork. And replication is always going to happen directionally towards the replication fork. So we're looking at a direction of the copy of DNA being made going this way. We are going this way, along with helicase this way. So we'd call this one the leading strand. Well, why is it the leading strand? Well, let's take a look at the strand here and what directionality it has. We're saying that this point is the 3 prime end. So we are going towards the 5 prime end. That's 3 to 5. Well, keep in mind here, guys, and this is where people make a mistake sometimes, is that the DNA polymerase can only add in a 5 to 3 prime direction. And because DNA is anti-parallel with one another, you must have this new strand that's being added. It must be the opposite of the template strand down here. The template strand is 3 to 5. It is called the leading strand because it's now being added, the new complementary sequence is being added 5 to 3. And that's what polymerase does. It works in a 5 prime to 3 prime direction. So this one is adding in a continuous line towards the replication fork, and it's being added in a 5 prime to 3 prime direction by attaching to a strand that's going 3 to 5 towards the replication fork. That's a mouthful, and that's always something people struggle with. So please ask me questions if you have questions about it. The lagging strand is much, much more difficult. The lagging strand also has to work in a 5 to 3 prime direction. That's how the enzyme is added. That's how its chemistry works. It also has to work towards this replication fork. So because this original template parent strand up here is running 5 to 3 already, and that direction towards the replication fork, the template strand is already 5 to 3, that means that somehow we have to add 3 to 5 going towards the replication fork, but we can't add 3 to 5. So what happens is a new enzyme called RNA primase comes down, and it will start to lay down some template strand here of RNA. What that then does is it allows DNA polymerase, another version of DNA polymerase, to attach and to start adding in a 5 to 3 prime direction. Now the 5 to 3 prime direction here would be going backwards. See how it would be going this way, away from the replication fork? This is the complex part. It's going to lay down a primer. It is going to work this way because it has to add in a 5 to 3 prime direction. But it also needs to accomplish the goal of going towards the replication fork, which is going this way. So it lays down a track. It then adds its bases going this way. It then has to jump backwards like this, and then it has to go synthesize this way. When it's done there, it detaches. It goes backwards over here. It adds this way. So you can see how this is adding still in a 5 to 3 prime direction. The bases, sorry guys, still adding the bases in a 5 to 3 prime direction, but still the overall progress is making its way towards the replication fork. We're always going this way, but the bases themselves have to be laid down in this 5 to 3 prime direction. You then have another player here, another enzyme named DNA ligase. What I would like you guys to associate with ligase is glue. It sticks fragments together. So as this is added 5 to 3 prime towards the replication fork this way, and DNA polymerase comes along and adds its bases, it's going to let go, and there's going to be these little gaps that need to be glued together, these backbones that need to be glued together. Phosphodiester bonds need to be formed here. In order for that to happen, DNA ligase comes along and it seals them up, and it makes a positive, strong covalent link there in phosphodiester bonds in the backbone of this nucleic acid. Those segments, these little segments, are one of my most fun things to say in biology, Okazaki fragments, named after the husband and wife couple, husband and wife pair that founded them. Thank goodness they were both named Okazaki. That's a fun thing to say. It's always going to make its way towards the replication fork, but it must be added in 5 to 3 prime. That's why I said lagging strand, pain in the butt. If you watch the crash course videos that are embedded in the IQ, also you'll see Hank Green also calling this thing a major pain in the butt, the lagging strand. This is just an overall image of what we just said. I'm not so concerned on you guys knowing what DNA polymerase 1 does versus DNA polymerase 3. Your book certainly will make mention of it. In the leading strand, it uses DNA polymerase 3. It allows for this continuous movement. And the lagging strand, DNA polymerase 1, is used to lay down bases from the RNA primer. That's farther than what we need to know. What we need to know is that DNA polymerase adds specific letters, and that it does make mistakes. And sometimes those mistakes, should they lead to a trait that allows for variation, which would cause a beneficial phenotype, then that would be enacted upon, and that allele, then that change in gene due to that mutation, would be selected for in a population if it was given an advantage. Okay, this is very complex, guys. It's one of the tougher things we do, and I understand that. So we have to take our time with it. And we're also moving along. So I want to remind you, one of the reasons why I'm shooting these videos and we're walking through this is so we can spend some time having conversation in class and in lab instead of me just yapping at you, because it will get really, really boring if I was just doing this in person. And I'm also long-winded, and I think you guys have caught on to that. So I have a 15-minute limit here, which I'm approaching right now. And I have to just sort of get straight to the point. So rewind it, annotate, take notes. I will post the slide notes here in a second, so you guys have access to those. Come to class tomorrow with questions for me, guys. Okay, try to chew through this on your own. Just do part 1. I hope you guys have a nice night, and I will see you tomorrow in 3rd and 4th period.